On the contrary, what I would like to do is add one more word of thanks to what Nir has already done, and that is to thank the audience. Thank so many of you for coming and for sticking it out to the end. I hope not the bitter end, but the end nevertheless. I will speak to you today about work which is done by the colleagues listed on the first slide. There was a pointer here. I'll speak to you about work which is all related but along four lines. In fact, the first bit, which I'll touch on only briefly, is really a postscript to what Nir, who was a participant in the work, presented so beautifully yesterday. After that, work concerning transcription again, though not in the context that Nir did of a naked promoter, but rather a chromatin template. After that, the mechanism by which that template is formed, and finally, the state of that template in whole cells. Three parts of this work, I should say what's at the top and at the bottom was published in the last couple of months. The part here in the middle just a few months ago, and this part not yet published, though not for want of trying. Now, I'll also begin with a slide similar to what Ada showed to introduce her work, only call your attention to one point in particular. So everything I have to say relates to transcription by the enzyme RNA polymerase II, one of three polymerases RNA polymerases in eukaryotic cells, but the one solely responsible for synthesis of the messenger RNA, leading to the synthesis of proteins. And as the first step in the pathway of gene expression, POL2 transcription is a focal point of cellular regulation. It's the end point of a great many, perhaps most, signal transduction pathways. And especially for the purposes of what I have to tell you later, the intricate regulation of POL2 transcription is key to cell differentiation and development. Now, we and others over the course of the 1980s, and I relate this in the spirit of the historical accounts you've heard from others and the school which this is in part, as well as a conference. We, in the course of the 1980s, identified a set of proteins that are required for accurate initiation of transcription on a naked promoter DNA molecule in vitro. Of course, the polymerase, which, as you'll see shortly, in a crystal structure unwinds the DNA double helix and is capable of RNA synthesis. Then a set of additional molecules, because the polymerase alone is not capable of recognizing a promoter, not capable of synthesis of the first couple of dozen phosphodiester bonds. For those essential functions, it requires what we call general transcription factors, and they go by the letter names B, D, E, F, and H. Polymerase itself is made of a dozen polypeptides. The general factors comprise 25 proteins, but still that's not enough. It was at first thought this constellation of molecules was complete, that it was capable not only of promoter recognition and the initiation of transcription, but also of a response to regulatory influences. In the early 1990s, a group of my colleagues discovered an additional assembly of proteins required for regulation. We refer to it as mediator because it goes between regulatory proteins such as gene activator and also repressor proteins and the machinery that I have just described. Mediator is also a large assembly of proteins. In yeast cells, 21 proteins with a total mass of 1 million Daltons. In human cells, the same 21 proteins and often additional molecules depending on the cell type. So the picture to this point, and the one which has, as I say, emerged through the course of the 80s and 90s, what is shown here, some 60, nearly 60 molecules assemble in a complex comparable in size to a bacterial, or a bit larger even than a bacterial ribosome, at every RNA polymerase II promoter prior to every round of transcription. For structural studies, we began with the polymerase. For various reasons, it might have seemed appropriate to begin with the smaller, simpler general factors, but in many cases, they only adopt their fully folded structures in the context of the assembly with RNA polymerase. At any event, I'll only show you one picture of the structure of polymerase because it's needed to understand the one important point I want to make relating to the structure that Nir presented before I go on to the rest of the lecture. And that in turn really is a kind of starting point for the rest of the lecture. So this is one view of the structure of RNA polymerase in the form of a transcribing complex with both DNA and RNA present, solved in 2001. And here what you see, of course, is the polypeptide chain and also strands of the DNA and RNA modeled into the electron density. And what the picture reveals, in this direction of view, the DNA double helix is seen to enter from the right. It unwinds 3 residues before the active center, after which there is a sharp bend, in consequence of which the axis of the entering DNA double helix is at nearly right angles to that of the exiting RNA-DNA hybrid helix. This region, this unwound region, commonly referred to as a transcription bubble. The reason for the sharp bend is because of a wall of protein density that blocks straight passage through the polymerase cleft, and it serves a deeper purpose that I could comment on later if anyone is interested. Finally, I will turn this off. Finally, to the structure which Nir presented yesterday, let me begin by explaining that we tried for many years to enlarge this picture by the addition of one or more of the polypeptides of the general factors and crystal structure determination. We were successful with only one of the 25 polypeptides, and we were never able to form a stable complex of the others. And it became apparent that it would be necessary to put Humpty together again, that is, to assemble all the parts from what had broken down in the course of isolation, to obtain a stable entity whose structure we might investigate by what I will tell you, what Nir mentioned yesterday, cryo-electron microscopy, or we hope eventually also by crystallography. Now, there was a major impediment to assembling all the components, and that is one of them, factor H, which is made of 11 polypeptides and is itself over 500,000 molecular weight, could never be isolated in a meaningful quantity in a pure form. It turned out that was because of, again, a biological reason we came to understand, and then Kenji Murakami constructed a mutant of yeast that overcame that difficulty, enabling finally access to sufficient quantities of all the factors, including H, for assembly of the complete complex, which was itself a challenge which he, Kenji Murakami, overcame, that led to solution of the structure which Nir showed, in which I will present only to make one additional point beyond what he has done already. So this is a picture you may recall from what he presented yesterday, and it shows the structure of the pre-initiation complex before the formation of what I showed you from crystallography a moment ago. It consists of the polymerase and the general factors, the polymerase here in gray, the general factors in blue, pink, aqua, yellow, and then the DNA as well, apparent at the resolution of this analysis. You can see that the crystal structure of the DNA that I presented a moment ago fits well into the electron density, the envelope derived from cryo-electron microscopy. Nir mentioned that the structure in the region of the mass of the general factors is less well resolved because of motion in that region, a problem which we have not yet overcome. Nevertheless, the DNA is clearly visible. The double helix, even the grooves of the double helix, are plainly apparent, and that leads to only 2 comments that I wanted to make, as I say, to add to what Nir did, that are relevant to what I have to tell you later on. So the first remark is the path of the DNA here in the promoter before melting to form the transcription bubble that you saw in the previous slide, before melting produce single strands, is not perfectly straight. There are actually 2 sharp bends one about 30 base pairs downstream from the so-called TATA element or TATA element of the promoter, and another about 45 base pairs downstream. This bend in particular serves an important purpose, which I won't take time to describe now, but I brought a couple of extra slides and I'd be happy to explain why that bend is so significant later on. The point I especially want to make about the path of the DNA is surprising, and it's shown here by a view down the axis of the double helix. So as it were, in this viewing in this direction from the previous slide, the DNA then appears mostly green, and the striking observation is that promoter DNA does not interact at all with RNA polymerase II in an RNA polymerase II pre-initiation complex. It is only in contact with the general factors. And from that emerges a principle, and we can, I think, readily understand why. And it is the promoter DNA before melting is relatively rigid and must follow an essentially straight path. But then, as I showed you previously from the crystal structure of the transcribing complex, DNA must bend through about 90 degrees in order to interact with RNA polymerase. So what we understand is the general factors serve the role of recognizing the promoter, bringing it to the polymerase, and then by action of one of those polypeptides, which is an ATP-dependent DNA helicase, melting the DNA, after which the single-strand region can bend, the DNA can enter and bind in the Pol II cleft to support transcription. Now, I call your attention to the mode of interaction of these proteins with DNA for one particular reason that is what leads to the rest of what I have to tell you this afternoon. It would seem obvious from the picture that I've presented, and indeed we have always imagined that the RNA polymerase II transcription machinery forms on a naked DNA, so on promoter in the absence of any other proteins, and in particular in the promoter in the absence of histone proteins in chromatin. I mean, we all know that DNA in cells is almost entirely associated with histones in the form of chromatin. And so it was imagined that the histones must be removed to enable transcription. And that is really the point that I will address in the remainder of what I have to tell you. So it was, I should say, apparent when many years ago I put forward the idea of wrapping DNA around a set of 8 histones in the nucleosome, that such wrapping would interfere with many DNA transactions, including transcription. In about 13, 14 years later, Yali Lorch showed that indeed assembling promoter DNA in a nucleosome would prevent the initiation of transcription by RNA polymerase II in vitro. And shortly after that, Michael Grunstein and colleagues showed that nucleosomes interfere with the initiation of transcription in yeast cells in vivo. And from this, we and Michael Grunstein arrived at the conclusion the nucleosome performs a regulatory as well as a structural role. It serves as a general gene repressor. There is no doubt of it. It actually is responsible for turning off transcription of all genes in all eukaryotic cells except those whose expression is brought about by specific positive regulatory mechanisms. And all the work since that time has been devoted to the discovery of those positive regulatory mechanisms. Now, I think the first indication of a solution of the problem came from genetic studies done by the late Ira Hershkowitz, a marvelous geneticist who died at an unfortunately very young age. And in what I think was a brilliant piece of work, Ira deduced the existence of what we now call a chromatin remodeling complex. He discovered what he called switch genes, and he inferred from his genetic analysis that they interact in a complex and that they are responsible for the relief from repression of transcription by histone proteins in vivo. Some years later, one of my students, Brad Cairns, succeeded in isolating the assembly of products of switch genes, discovered it's larger even than was anticipated, contains many more proteins, and in particular a switch SNF associated protein, SWAP73, that Brad realized had a homologue in yeast, and on that basis, pursuing isolation of the homologue in its native state, discovered a much larger complex made of 17 proteins that we refer to, whoops, why did that happen? Oh, an update is available for my Adobe Reader. No, that wasn't. Somebody have a better idea? Okay, so as I say, Brad discovered a 17 protein complex, which is shares a lot of similarity with switch-SNF, but there are important differences. The switch and SNF genes are not required for cell growth. They may be deleted. And there are very few copies of the switch-SNF complex in yeast, and of its counterpart, I might add, in higher cells. RISC, on the other hand, is essential for cell growth. The genes are required for life, and there are a large number of RISC complexes in cells, on the order of 1,000 in yeast and more in higher cells. Both switch-SNF and RISC contain a subunit which is an ATP-dependent DNA helicase, and more specifically a translocase, with the consequences that are illustrated in the next slide. So RISC in the presence of ATP can both slide a nucleosome, that is to say, produce translocation, and in that manner it can expose a site for binding a protein, RNA polymerase, or what have you. We've since found RISC in the presence of ADP can also remove the histone octamer from DNA, and in that manner equally expose a site for interaction with a protein that needs to make direct contact with the DNA. And on this basis, you might say that the problem of relief from repression by the nucleosome is solved in principle. But of course, it's really only a formal solution to the problem, and it remains to establish whether this is all that's required, or whether there are additional players, or whether this is really relevant at all to the problem of transcription. And so for this purpose, others over the years have formed templates for transcription upon which they assembled purified histones to form nucleosomes. And they found, of course, that, as we had mentioned done before, as Yali had shown, that the histones would interfere with transcription, and then in the presence of a remodeling complex and sometimes an additional protein, they could bring about partial relief from that repression. I felt for many years that it was essential to perform such a study on a gene in its native state, that is to say, isolate a gene as it occurs in nature, and find out what is required to elicit transcription with a defined set of proteins in vitro. That was a problem which occupied us for many years. The approach that we have taken to solve the problem is illustrated here. So we place sites for recognition by a recombinase upstream and downstream of a gene in yeast, and in this case, the same gene as Michael Grunstein used to demonstrate the inhibitory effect of a nucleosome on transcription, the so-called PHO5 gene of yeast. And then we induce expression of the recombinase that recognizes those sites in the yeast cells in vivo. That brings about excision of the gene in circular form as chromatin, but now released from association with the majority of the yeast genome. And in that form, the gene as chromatin is soluble, can be isolated, and we did so by an affinity method. We could purify this circle 200,000-fold to virtual homogeneity. Now, the purification to virtual homogeneity was a kind of Pyrrhic victory, because on the one hand, we had now a suitable substrate. On the other hand, we had almost none of it, because the gene occurs in a single copy. Any gene of interest to us for this purpose occurs in a single copy in yeast or other cells, which means that the quantities we obtained were at the attomolar, 10 to the minus 18th molar level, which posed a real challenge for the detection of transcription, which was surmounted by Shigeki Nagai in the work that I will now describe. And so Shigeki began with the set of proteins that I have already mentioned, polymerase and the general factors. When applied to naked DNA extracted from the chromatin circles, he was able to detect, by a means I won't have time to describe, accurately initiated transcription even despite the small quantity of material. When applied to the circles in the form of chromatin, he recapitulated Yali's result, namely the absence of any transcription. This was all, of course, as expected. He went on through the course of many experiments to discover what constellation of molecules is needed to elicit transcription from chromatin. And these begin with the PHO4 gene activator protein for the PHO5 gene of yeast. What had previously been regarded as an elongation factor, but we now know is also required for initiation, and there's supporting genetic evidence from Fred Winston. Mediator that I've mentioned. A complex of the so-called TAF proteins and some additional important molecules referred to as SAGA. Those additional important molecules include a histone acetyltransferase responsible for histone acetylation. That protein is, in the case of SAGA, GCN5. And then one more protein that I'll come back to later called SGF29 that recognizes a particular histone mark, in this case methylation of a histone, that is also relevant to transcription. And finally, in these experiments, switch SNF was used, but RISC could have been substituted and would perform equally well in such a reaction. Now, what Shigeki found then is, again, he could detect transcription from DNA extracted from the chromatin circles, but more to the point, in the presence of this set of additional molecules, and in a reaction that requires all of those additional molecules, finally he could observe meaningful transcription from the gene in the form of chromatin. Now, because of the small quantity of material and the elaborate means of detection, it's necessary to establish specificity. It was done in various ways, including what is shown here, a single base change in the TATA box of the promoter, which completely abolishes chromatin transcription. You will doubtless have noticed we didn't find transcription almost as good as that from a naked DNA. The surprising finding, transcription from chromatin was much better than that from a naked DNA molecule, utterly unexpected. And as the next slide shows, as the concentration of all the proteins present in the reaction is diminished, the ratio of transcription from chromatin to that from a naked DNA molecule increases, from which we infer those proteins must interact with chromatin, make an interaction they can't do with DNA. And that is the basis for this remarkable enhancement of transcription by the presence of histone and other proteins on the DNA. Now, just to return to what I said a moment ago, I alluded to the presence of the histone acetyltransferase in the SAGA complex. As you might imagine, its substrate, for the purpose of acetylation, acetyl-CoA, is required in the reaction with an optimum of about 5 millimolar. And then I alluded to the presence of the SGF29 subunit of SAGA. That subunit recognizes the so-called H3K4 methyl mark, which is to say methylation of lysine 4 of histone H3, associated, I might add, with the promoters of all genes active in transcription in all eukaryotes. Deletion of that subunit from SAGA significantly impairs, though does not abolish, transcription. So finally, just to summarize what I've told you to this point before I go on with more details, what is remarkable, the level of transcription from chromatin was, in the experiments we have done, at least 20-fold greater than that from naked DNA, and we anticipate even more so if the concentration of proteins could be reduced still further. And because of the dependence on concentration, we infer, as I have mentioned, interaction of the proteins with the chromatin, or more specifically, and I will show you more directly, interaction with nucleosomes. And lastly, to repeat what I said only a moment ago, this interaction evidently involves histone modification, both acetylation and methylation. Now, the question naturally arises, which nucleosome of the gene in the form of chromatin is the target of this interaction? And we can answer that question by taking advantage of findings made many years ago by Wolfram Hertz and his colleagues. So they showed that the PHO5 gene in its repressed state is, as you might imagine, packaged in a continuous array of nucleosomes. And one of these happens to cover the PHO4 binding site. Another covers the TATA box of the promoter and the transcription start sites. Hertz and colleagues also showed that on induction of PHO5 gene transcription in yeast, so conversion to the activated state, 2 nucleosomes are removed, exposing the PHO4 binding site. The nucleosome over the start site is not removed, but only caused to slide, exposing the TATA box, but still covering the transcription start site in the activated state. So we could take advantage of this to make another chromatin circle, only in this case a much smaller one. We place the sites for recombination immediately upstream and downstream of the promoter. We isolate a circle which in the repressed state contains only 3 nucleosomes, and in the activated state contains only a single nucleosome positioned over the start site. And Shigeki found that transcription was still enhanced by the presence of that single nucleosome over the start site. Transcription from that template markedly greater than that from the corresponding naked DNA molecule. So from this we have come to believe that a nucleosome covering the start site, what is referred to as the +1 nucleosome of all genes, is actually not repressive, but rather required for the initiation of transcription. Now, the PHO5 gene of yeast has long been thought of as a special case. But what I would like to emphasize to you, it is not a particular example, but rather represents a paradigm for all of eukaryotic transcription. The pathway that is shown here is, I believe, completely general, and I'll show you evidence to support that idea. So one begins in a tissue where a gene is never expressed, and we know this from work of many people over the years. A gene is packaged in a continuous array of nucleosomes. And as I'll now explain in more detail, we know that in tissues where genes are expressed, they take the form that is shown here, and I'll show you supporting evidence for that coming from studies in yeast. So here, what you see is a map of the locations of nucleosomes produced by digestion with micrococcal nuclease between the particles and then high-throughput sequencing across a 100 kilobase region, 100,000 base pair region of one of the yeast chromosomes. So what is plotted is the likelihood of finding a nucleosome as a function of distance across that region. And what you see, a more or less monotonous array of nucleosomes punctuated by apparent absences. And if we zoom in on one of these, what you see, those absences correspond to the lack of a single nucleosome and the presence of an adjacent, rather strongly positioned particle that lies above a transcription start site. These bare regions, which are called nucleosome-free regions, or NFRs, contain the regulatory elements and the TATA box of the gene, but not, of course, the transcription start site. It has been thought since these were first discovered by Ali Rando and studied by others that their existence is due to an abundance of AT-rich DNA in the NFR region. And it was known long before that poly-DA, poly-DT DNA is refractory to the formation of nucleosomes. Yali Lorch and colleagues in the lab have investigated this question and discovered quite to the contrary. The presence of AT-rich DNA is not directly responsible for the absence of a nucleosome, but rather does so through the agency of risk. So Yali assembled a nucleosome on NFR DNA and on that of the adjacent nucleosomal regions, measured the rate of removal of a nucleosome by risk, discovered that it is rapid in the case of the NFR, but virtually absent for the adjacent region. And she showed that the basis for that is what is shown here. So here is one of the rate of removal or the time course of removal of a nucleosome from one of those adjacent regions in the absence and in the presence of a 7 residue stretch of oligo-DA, oligo-DT inserted in the nucleosome. And you see that promotes in a dramatic way the action of risk to remove the histones from the DNA. So I'll summarize what I've told you to this point in a couple of ways. One, we've expanded our picture of what is a pre-initiation complex. We now believe that it consists of 99 proteins with a total mass, if Otto will pardon, larger than the largest ribosome, in excess of 5 million Daltons, which includes not only polymerase general factors, but mediator, saga risk, and especially a nucleosome. I might summarize in another way by saying that it enlarges our picture of the functional role of a nucleosome. So I told you that Yali discovered years ago that it plays a crucial negative regulatory role. Now we know it is equally important for the function in transcription. It is recognized by the transcription machinery. Now you might ask, well, why did nature do it that way? And I think the answer is straightforward and very beautiful. It provides a fail-safe mechanism. So even after a gene is exposed and the regulatory elements are available, something further is required to initiate transcription. You don't make a fatal mistake and transcribe the wrong gene in the wrong place at the wrong time. You have to have a nucleosome still present over the start site to prevent it happening unless regulatory proteins bind in that exposed region, bring about the formation of a pre-initiation complex to start the reaction. Now the last thing I will tell you in this final chapter of the talk I give this afternoon concerns the structure of chromatin as it relates to transcription beyond the nucleosome. After all, there is more to the structure of chromatin than the single particle that resides on the transcription start site. We know that, as I've already mentioned, the entire DNA of the organism is packaged in an array of nucleosomes to form what people call chromatin fibers that are then further coiled or folded in some manner. In what manner? And what is the structure of a gene? What is the organization of a chromatin fiber of a gene undergoing transcription? That is the question. And the next slide really serves just to emphasize our ignorance. So this is a classical picture, probably dates back to the work of George Palade from 60 or 70 years ago, showing a thin section electron micrograph of a eukaryotic cell nucleus. It could be any eukaryotic cell nucleus. It's a picture that I used to show every year to medical students at Stanford when I taught medical histology. I don't show it anymore because the Stanford administration unlimited basic science from the medical curriculum in their wisdom. So now I have to be satisfied with imposing it upon a group like you. Anyhow, this is the picture, and as many of you know, it will characteristically show dense chromosomal material around the periphery and less dense material in the middle. And long, long ago, it was shown that the majority of transcription happens in the interior, in the region of less dense chromatin, classically called euchromatin, in contrast with the more dense stuff around the periphery, classically referred to as heterochromatin. In very recent years, just over the past decade, some progress has been made towards understanding the folding or the coiling of chromatin fibers in eukaryotic nuclei, and it's a result of a method that you have heard mentioned. I think Tamar Schlick mentioned it, called chromosome conformation capture, which, when performed with high throughput sequencing, is now called HI-C for the genome-wide analysis. And the method is extraordinarily simple in principle. It consists of fixing the material, usually with formaldehyde, so cross-linking proteins to one another, and then fragmenting the DNA. Two fragments which may derive from distant regions of the genome brought together by an interaction of proteins will exhibit cut ends near enough that they can be joined by ligase, and then detection is through high throughput sequencing to discover now the adjacency of regions that were previously, as I say, in remote regions of the genome. And the results of this kind of analysis, published by many groups over the last 10 years, are the sort shown here, and this is from work of Job Decker and his colleagues, but it could come from many other papers. And what is characteristically observed, there are regions where a point, so here the X chromosome of a human cell is shown plotted on both axes, and a crosslink can be detected between one point and another which lies a megabase away. And the resulting picture is of blocks of so-called topologically associating material or topologically associating domains, or TADs. So one sees blocks interspersed with regions of little such long-range interaction, a block another point where there's little such long-range interaction, and so forth. It wasn't apparent, and there's been a lot of question about the significance of these observations, and many people who don't actually make the measurements themselves doubt their significance, because one is looking, after all, at fixed material, and you really can't know how that interaction came about. And it was suggested perhaps this represents looping for regulatory purposes, or perhaps it represents transient interactions of material that is mobile in the nucleus, or what have you. We could make progress, gain some insight through a series of experiments, which are the last that I will describe. It occurred to me that such a pattern of blocks of apparently self-associating material along the length of a human chromosome was reminiscent of a classical picture, a picture from classical cytology. This is a picture that was first produced in the 1930s of so-called polytene chromosomes of Drosophila melanogaster. So it was discovered that in the salivary glands of dipteran larvae, chromosomes may be replicated to produce as many as 1,000 copies without cell division. And those many copies reside in the cell nucleus perfectly aligned with one another, in consequence of which one sees an alternation of because this is a reverse or negative of the original picture, actually dark, light, dark bands along the length. And such a band pattern was really the basis for understanding gene deletion, gene duplication, genetics at the level of chromosome structure in a general way for decades until DNA sequencing became available. So the question we asked, does the band pattern in a polytene chromosome relate to the pattern of TADs revealed by Hi-C? And Kyle Egan and Tom Hartle performed Hi-C analysis on dipteran salivary glands, dissecting hundreds really of glands for the purpose, and they discovered that TADs could be detected, and in particular in the small number of cases where the DNA coordinates of the bands of polytene chromosomes were known, they could produce the picture shown here. Here, the coordinates of the bands that are known are lined up on the left side. Of course, the bands contain varying lengths of DNA. In red shows the overlap of the band DNA with the results from the Hi-C analysis, the TADs. In black, regions of non-overlap. And of course, what's striking is that the bands really do correspond to TADs. Michael helped us with the statistical analysis, and he will vouch for the validity of the analysis. Now then, as I mentioned, one only knows the DNA coordinates for a relatively small number of bands, or did in the past. But we know from the Hi-C analysis the precise DNA coordinates at every point along the genome. And we could do the reciprocal study. We could start from the TADs and inquire about their relationship to bands. So what I'm going to show you is one of many examples, but it relates to the band that is indicated here by the arrow. I don't know if you can see that very well. Maybe if you dim the light for me. I think you can see that band a bit better. So yes, so now you can see that there is a band at this point. And then what Kyle Egan did was to produce fluorescently labeled complementary DNA from the points on one side or the other of a TAD and perform fluorescence in situ hybridization or FISH analysis. And you can see that the DNA from one or other side of the TAD lights up the interbands on either side of the band. And then similarly make a FISH probe for the DNA in the middle of the TAD, and it lights up the band itself. And he's done this for quite a number of bands. You can turn the lights back up. And we conclude then that not only do bands correspond to TADs, but TADs correspond to bands, and there is really a perfect one-to-one correspondence. One could go on to ask whether this pattern that is seen in polytene cells of dipteran larvae have anything to do with the organization of chromosomal material in the nuclei of any other Drosophila cell. And in fact, many high-C analyses have been performed on Drosophila embryo tissue, on Drosophila tissue culture cells. And here what was done was to compare the pattern of polytene TADs with the pattern of TADs in diploid cells of Drosophila, in this case tissue culture cells. In green are regions of overlap, black non-overlap, and you can see that there's a predominant close association from which we may conclude that not only are bands equivalent to TADs, but polytene TADs really reveal the organization of the chromosomal material in the diploid nucleus. So nature has made us a marvelous gift. It has given the opportunity of seeing in the light microscope what is the pattern of organization or of condensation of the chromosomal material in the diploid nucleus. And then on that basis we can simply put a ruler to the light microscope picture that I showed you of a polytene chromosome before and determine what is the degree of condensation at any point in the genome. And when that is done, the results are expressed in terms of what we call a packing ratio, which is the length of the DNA that we know from the DNA sequence analysis, divided by the length of the band or interband, the length of the corresponding chromatin fiber as it is revealed in that light microscope picture. We start with the interbands. There we can estimate a packing ratio of between 5 and 10. When I, many years ago, put forward the idea of a nucleosome, I could calculate what would be the expected packing ratio in consequence of wrapping DNA around the histones alone. Could calculate that the packing ratio for a chromatin fiber, for an extended fiber, the number I calculated was 6.8. Clearly what we see in the interbands are extended chromatin fibers. Now we know from the DNA sequence analysis that the interbands contain the regulatory sequences and the promoters of genes. It was shown by in the original cytological analysis, there are two kinds of bands, light bands and dark bands. The light bands have a packing ratio a good deal higher than that in the interbands, up to about 10-fold more condensed. We don't know the pattern of coiling, but what is important, they contain the gene bodies of all the active genes. And finally, the black bands have a packing ratio which is about 30-fold greater, up to 30-fold greater than that of the extended fibers, and they contain what is transcriptionally inactive material. So you will recall this corresponds to what was called euchromatin, active gene regions, and heterochromatin. So finally, the analysis that I have just described gives some color to the classical picture of the interphase nucleus. Now we can say that in the transcribed region, the promoters, the regulatory regions and promoters of genes reside in fully extended chromatin fibers. They reside in a structure that we know. The gene bodies in a more condensed fiber whose structure we have yet to determine, and inactive genes around the nuclear periphery in a still more condensed state. And I will just conclude with these last summary remarks. So I've told you from the work of Yali Lorch, we know that an NFR is created by the action of RISC and ATP, and that it exposes regulatory and TATA elements of genes, but not the transcription start site. I've told you from her work and that of Shigeki Nagai that we know the nucleosome performs a dual functional role. It is a general gene repressor, but also required in every instance for Pol II transcription. You will recall that after the general factors interact with the promoter, they serve the purpose of not only delivering the straight DNA to the Pol II active center, but melting it so that it can bend and bind there for the purpose of transcription. And in the last part of the lecture I emphasized, we know the structure of the gene as it occurs in nature in the transcriptionally active state. Regulatory regions and promoters reside in fully unfolded chromatin fibers. And with that, I thank you for your attention. I'd be happy to take any questions.
Giton?
So how does the fully condensed areas open up?
So it is not known how they are folded in the first place, so no one has the vaguest notion how they're condensed, and so it's not known how they're decondensed. However, there are some observations that are made that are doubtless relevant. So there are particular histone marks that are associated with those constitutively inactive chromosomal regions. Methylation of the lysine number 9 on H3, methylation in other cases of lysine number 27. It's known in some cases what proteins associate with those methylated histones, and presumed that they perform a structural role bringing about that condensation. But how that happens, what is the condensed state is not known, and how it is reversed is not known. Mike?
Roger, in the euchromatin, you mentioned that it was a mixture of naked fibers and somewhat condensed fibers. Are there actually two phases, or is it more a continuous?
There are discrete regions. So it's clear from the light microscope pictures. Those gray bands have discrete boundaries. So I think that answers your question.
Well, but where are they? Are there three kinds of bands then, or two kinds?
Two kinds of. There are three kinds. There are interbands.
Okay, fine.
And two kinds of bands.
Okay, fine.
Or if you like, three kinds of bands would be more accurate.
Okay.
Tom?
If I would be looking at the Eurochromatin in an interphase cell that is a single chromosome, right? This is not the example that you presented. Would the organization be similar?
So we believe from the virtual identity of the distribution of TADs, or the pattern of cross-linking between the polytene state and the diploid state, that the answer will be yes, they are the same. The only difference is that in the polytene state, they're fully extended, they're stretched out because the chromosomes are aligned. But of course, in the diploid state, that can't happen, and I might just possibly have, there he is, a picture to illustrate that point. So in the, what happened to that? So in the interphase nucleus, where in contrast with the polytene cell, such alignment is not possible, or such a straight or linear organization cannot occur, then one must have the condensed regions, as we know, associated with the periphery, another condensed region associated with the periphery, and then it's unavoidable that the regions between them form a loop that extends into the interior of the nucleus, which contains both the gray bands and also the interbands, the region that is regulatory and also contains start sites.
But let's say in the picture that you showed that EM.
Yes.
The
This would be the
cell.
same as that picture.
Right, but
with
there's an issue of scales, right? Because you have the heterochromatin, which is close to the nuclear membrane.
Which
was
is
also
here.
There. The distance is gigantic going into.
but these include fully extended fibers. Think about it.
No. So I was
And also only partially folded fibers, at most tenfold condensed.
and where is the transcription happening?
Transcription is happening in the interior, and more particularly, as I mentioned, the promoters lie within the extended region adjacent to these partially condensed regions.
And do you think that every nucleus has the same super
I'm sure of
structure?
it. I'm
or
sure of
different?
it, yes.
Why are you so sure?
Because the analysis that we did in Drosophila shows the same result, the same pattern of TADs, and so the same apparent organization that one finds in the polytene nucleus, also in cells grown in culture, also in embryos, which are composed of, as you know, very many tissues. So there are some discrepancies, which is hardly surprising. For example, there are some cases of looping important for regulation, which cannot occur in polytene chromosomes because they don't bend back upon themselves, but which demonstrably occur in some differentiated cells for purposes that you're probably familiar with. It happens in human cells for turning on one or other appropriate receptor gene in an olfactory tissue or in immune tissue, or what happens. Those kinds of long-range interactions, which bring about looping that can be detected by high-C analysis do not occur in polytene chromosomes and are obviously a special case.
May I allow one small bit? So, how the various chromosomes don't get entangled?
So the most likely, first of all, the most powerful, as it were, untangling mechanism is topoisomerase II, which is not only very abundant, but even a component of the most condensed states of chromosomes and allows strand passage of DNA. So in effect, makes one region of DNA invisible to another, so that tangling would not be expected to occur in an irreversible manner. Now, of course, it also would enable tangling, except that as chromosomes condense through a mechanism that we have not yet discovered, untangling will occur as required to achieve the final state. Bear in mind the TADs, what our analysis has shown, and I failed to emphasize, what we learned from the analysis of polytene chromosomes, TADs represent stably condensed states of chromosomal material. And in fact, the axial condensation, which is apparent from the polytene chromosome, pertains even to the TAD itself, something which we're now analyzing in finer detail.
Sorry. Hi. I have a question about the peak. So, more or less, how do you think it relates the structure of the peak with the ones with mediator from the Kramer lab? So, is there any big structural rearrangement, or it's only a DNA that changes the position and it's now inside the polymerase? Or
Right. So, Patrick Kramer has produced a cryo-EM picture showing the association of part of the mediator complex with part of the general factor complex, actually a much reduced version. Doesn't include any of what I call the G lobe, that very large bit on the top. We have, and I should say in particular Philip Robinson, has solved the entire structure, including the entire mediator and the entire pre-initiation complex, polymerase with all the general factors. His picture is consistent with that determined by Patrick Kramer and colleagues, but of course adds all of the rest of the detail.
This TAD structure is right, a function of the cell stage, right? Cell cycle stage. Can you well,
yes and no.
interphase, metaphase?
Everything here applies to interphase.
Right. So, but could you imagine
What happens in metaphase?
Yeah, and can you imagine techniques like time-resolved crystallography, analogs of this, that one day could visualize the dynamics of these TADs?
Yeah, okay, I can imagine it. Wouldn't I like to do it? So there was one high-C study performed on metaphase chromosomal material, and it gave a perfectly uniform but rather fat diagonal, which I think actually reflects, as it were, the uniform axial condensation along the whole length without any interbands anymore. The authors of that paper actually said they thought what happens is the whole thing unfolds and then refolds in a different manner, which seems rather wasteful. I can't imagine nature would do it that way. So we'll find out eventually whether the TADs are conserved in the metaphase state. And if so, whether the interbands, the extended regions, then themselves become TADs, so to speak, with a similar packing ratio before finally one arrives at the full degree of condensation in the metaphase state. But to answer the other part of your question, sure, one would like to use microscopy such as Tommy Kirschhausen described and other such methods, perhaps crystallography if it were applicable, but so far has not been successfully applied to the most condensed states, to reveal this process, not only as it happens, but to understand what are the intermediate states and what is the trajectory of the chromatin fiber in the final folded state, which is really the goal of all of the work on these lines.
Ila?
So I have a general question about moving from basic to applied science in the transcription world. So last week in Nature, Bradley Bernstein showed that in gliomas you have a frequent mutation in isocitrate dehydrogenase, which causes a problem with essentially with DNA methylation dissolving and a insulator protein that cannot bind, moving a enhancer to another promoter, upregulating a receptor tyrosine kinase and causing division in gliomas. And they did succeed to reverse it with a general DNA methylation dissolving, but it's not specific. How much do you think, from your understanding of the transcription world, we will be able to manipulate things in such cases and other cases of diseases which involve a wrong enhancer, promoter, and transcription interactions?
So as your question implies, there are some observations and ideas that relate to what sets the boundaries of the bands or of the TADs or of regions of the chromosomes divided up in the manner that I've said and that you and that Bernstein has shown, who's done very extensive analysis of this kind. There's a protein called CTCF, and there's a counterpart in Drosophila that probably plays such a role. Kim Naismith has an elegant idea for how condensed regions may be formed by what he calls an extrusion mechanism of DNA being powered or translocated through a complex that includes CTCF. So in the first place, there are some useful ideas and a starting point from an experimental basis. Those kinds of experiments and observations have not so far been connected with patterns of modification of histone. And so it's not yet known in a very precise manner how they relate. However, it's noteworthy that there are particular acetylation and methylation marks that occur in a redundant manner over long regions. And while this hasn't been emphasized, it seems to me unavoidable that those relate to structure. Eventually that will be found. It is remarkable that histone deacetylases have proved useful in the clinic. I mean, it's surprising that they prove to be somewhat efficacious as anti-cancer drugs, but then as we know, you don't need great specificity of an anti-cancer drug. Any poison will do, which is just a little worse for a rapidly dividing cell than for the rest of the body. And so HDACs probably fall in that category, histone deacetylases. The question that you ask about specificity is going to require discovering what it is that sets the boundaries of particular genes and divides their, as it were, regulatory and promoter regions from those that lie adjacent, what prevents a regulatory region from one gene from influencing that of a neighbor in the very experiment that you mentioned. That all remains to be
And
determined.
how general it is. I mean, I got to it only tomorrow. It will be in Haaretz. I helped to bring it there, but it seems like there should be some specificity that once we understand enough, we can deal with specific CTCF, for example.
Exactly. It will be found and it will be sequence specific, and it just hasn't yet been identified.
Thank you.
Hi, I have a simple question. So, DNA from highly compressed form to like 100 nanometer, 30 nanometer fiber until like to the active form. Do you think the histone modulator, this histone modification factors play as a central player, or do you think there's multiple classes of decondensers that is important in this process? Are these process like discrete, or do you think they're like a continuous, more like a continuous process?
So Tamar Schlick mentioned or alluded to, I think you mentioned, Tamar, that it's no longer thought the chromosome coils in a 30 nanometer fiber.
And there's a cryostructure, I think.
Which is probably
wrong.
No, I don't think the cryostructure is wrong, only I think that it is a product of assembly in vitro, and it's revealed readily in the electron microscope, but probably not general and may not occur at all in vivo. It's really not known. The prevailing, if you were to ask most people who study the subject now, they would express doubt about the relevance of those observations to the structure of chromatin in vivo. But your question remains. Obviously, as I mentioned, we know that there is about 10-fold condensation of the bodies of active genes and about 30-fold condensation of the fibers in interphase of inactive genes, and we don't know the trajectories of those fibers. I would repeat what I said a moment ago. It's highly likely that both acetylation and histone methylation are involved in defining those regions, but then proteins that recognize those marks must be the ones that actually determine the structure. And we have one example of that. Michael Grunstein showed many years ago that the mating type locus of yeast exists in a condensed state where it is inactive as a consequence of a particular modification state of the histones in that region and their interaction with special yeast proteins. And I think what he demonstrated in that case is still the sole example, and it is a paradigm.
Perhaps a final question. Hi.
The stretches that you show there in thin, low condensation state, are these related to also to DNA replication?
The state that I describe is actually one in, as it were, interphase in the absence of replication, and one doesn't know except to say that regardless of whether the chromosomal material is fully extended or entirely condensed, it is still a substrate for the replication machinery. The heterochromatin is replicated almost as readily as the so-called euchromatin. So the answer is probably the picture I show you here pertains more directly to transcription than to replication.
Hi, I have a general question about this called TAT. The size of the TAT is about 1 megabase per long, right?
It varies, but
it varies.
Maybe
also,
on average, I don't know.
There are these trans or cis-active enhancers that can be like far away, but it can, if you have this kind of organization, would that actually restrict the formation of enhancer in a trans or cis way?
So those kinds of long-range interactions involve a different kind of chromosomal looping in contact from what is responsible for producing a TAT. A TAT is a condensed state of chromatin brought about by fibers coiling in a manner that we have not yet determined and don't even know how to determine. Those long-range looping events are something which we think are a simple consequence of interaction of a protein bound, for example, to an enhancer and a target protein or sequence, often at a promoter or adjacent to a promoter. And as I emphasized, those don't occur in the polytene state because the chromosomes are stretched out and they visibly do not exhibit that property. But on the other hand, we know they occur in many special cases, and I mentioned in answer to Tom's question the examples of olfactory tissue, of cells of the immune system, and there are many more examples.
Okay, I think because of time we will end here. Thank you, Roger.
And thank all of you.